University of Michigan at Ann Arbor, and I spent the summer doing research at the University of Texas, and I did computational studies of novel inhibitors of aldolase A using molecular dynamics simulations in the lab of Dr. Penyu Ren. So our REU was cancer research oriented, and so when cancer cells grow in solid tumors, they eventually grow at a rate faster than that of angiogenesis, and so they enter a state of hypoxia. In order to deal with this, they use hypoxia-inducible transcription factor, or HIF-1, to regulate growth and to moderate response by stimulating glycolysis and cell growth and angiogenesis. So you can feedback regulate HIF-1 by regulating glycolysis, which is the mechanism by which cells break down sugar and generate ATP and produce pyruvate for use in the citric acid cycle. So about halfway along the glycolytic pathway lies aldolase, which is an enzyme that breaks down fructose into two smaller components. Back in about the 70s, they started characterizing several different inhibitors. One of those was naphthalene-diol 2,6-bisphosphate, which we call here ND1. And this mimics the substrate, and it's been shown to act as an inhibitor in vitro, but in vivo it would quickly be metabolized and would be broken down before it could even reach its target. So our goal was then to develop these analogs that would also act as inhibitors but would not be metabolized quite as quickly. So we started by ND5, which, as you can see, the oxygens and the phosphates are swapped out for CF2 groups. And one of our collaborators started to do some early characterization experiments and found that it was actually a pretty terrible inhibitor. It didn't seem to be doing much of anything, and so they asked us to figure out why. So we developed two intermediates. We developed NDA, which is asymmetric, so there's an oxygen, and then there's an insertion of CF2 rather than a substitution, and NDB, which symmetrically does the two CF2 insertions. So our goal was then to better understand the binding and improve the activity. In order to compute binding for energy, we use something called the double decoupling scheme for molecular dynamics. And so we run our simulations using the Tinker software, which is developed by the Ren lab and the Ponder lab over at WashU, and the amoeba force field, which exceptionally depicts polarizability. So to compute the delta G binding, we use the hydration energy and the difference in energy between just the protein in water and the protein with the ligand bound into it. Once we had our simulations run, we had some data output, and we found that the delta G binding for NDB and NDA1 compared to ND1 were significantly lower, which is a good thing. That means that it has a much higher affinity. So then we were able to break this down and do the delta G compositions. And the traditional idea is that there's an enthalpic and an entropic component. And the relative contributions provide a little bit of insight into how these series bind. So while, for example, some might be enthalpically driven, others will be entropically driven. So NDB and ND1 are both enthalpically driven, whereas these are more closely entropically driven. Now, an enthalpic component is going to consist of a sum of smaller interactions, and in this case we were able to characterize interactions which are shown here in a heat map. Darker color indicates a shorter distance between the two atoms, so what we did is we tracked the distance over the course of our simulation and took kind of the mean. And interestingly enough, we found that in some cases where the phosphate and the CF2 group could be in similar proximity, in some of our weaker binding inhibitors, the CF2 group seems to be in a much closer proximity for an interaction than the phosphate group, whereas in some of our stronger ones, the phosphate was in a slightly better position than the CF2. So we were wondering if this would play a strong role, of course, in these enthalpic components, and so we broke these down by looking at the interaction energies of a free phosphate and free CF2 bonds. In order to do this, we took interaction energies with water, and so we computed the energy for that interaction between a hydrogen bonding, and we actually found that the phosphate interactions are significantly stronger by almost a factor of 10 than the CF2 interaction, suggesting that when the phosphate is in a better position for interaction, that would have a fairly strong energy contribution compared to a CF2 group. The final thing we considered was the solvent accessible surface area, which allows us to compare the shape and size changes in binding pockets as the protein is bound to different inhibitors. Because per the traditional induced fit model for inhibition, if you have your binding pocket, it will change slightly in shape and size, and that will actually affect the overall systemic delta G of the system. So we actually found that as you roughly increase the mean SASA, it's roughly associated with a decrease in binding affinity, with the exception of ND5, which we suspect is due to its size. So our overall conclusions from the work suggest that NDA and NDB would have strong potential as chemotherapeutic drug leads, as long as we can synthesize them.